So my good afternoon class, I started the recording just now, but we just started. So my question to you is, how many different types of chromosomes are present in a human cell?
46.
Loudly, please.
46.
46 different types of chromosome. All right, that's one answer. Who was that?
Nuffle.
Okay.
I think 24, 23 or 26.
Give me one number.
23. 23 pair. It's 23 pair.
All right.
Don't you get
Not 23,
from
but
your-
23 pair, which is 46.
Yeah.
So
Yeah.
let's say one student said 46.
Yes.
Said
23 pair.
And then?
Isn't it
23 pair.
some from your mother and some from your father?
Slowly, Nicola, you're saying something?
No, I was saying, don't you get some from your mother and some from your father?
Give me the number. Yes, of course, you're right, 100%. But
23,
Yeah, you get
23
half,
each.
half. Yeah, which would be 23.
Yeah.
23.
23 pairs.
23 pairs. Okay.
Yes.
The right answer is that in a human body, or in humans rather, if I just say humans, would your answer change?
If you just say human, no, it will be the same.
Same, right? So the different types of chromosomes that are present in human is 24. Why? Because 23 plus the Y chromosomes in male. So the total is 24. Any argument? Any discussion? So
Where does the extra one come from though?
Yes.
you have to listen to the question. My question was that how many different types of chromosomes are present in humans, right?
Okay.
I didn't define male or female, right?
Male or female, okay.
So if it was female, it would have been
It'll be an X, right?
22.
Yeah.
If it is male, then 24, because males
Of the
have
Y.
3X chromosome plus 1
Y.
24, right?
Okay, cool.
Any disagreement, any debate? So who was, so are you convinced? I mean, another way of asking the same question.
Yes.
Okay. Yeah. So, so that's why we go back to the basics, right? So when I started this course, I said that I will be talking about fundamentals of molecular biology, and then navigating into what's happening in the technology, right? And my simple philosophy is that you have the right knowledge when you go into the real world, right? And and you're competitive, you're strong, and so yeah. So this is a typical figure, right? Karyotyping. This is how the field of cytogenetics started, where a lab hematologist or a tumor biologist would look into the chromosomal arrangement and chromosomal length, and would work on with light microscopy, and few stains that would color the chromosomes, and many discoveries, particularly relevant to leukemia, were discovered by just looking at the blood cells and measuring the arms of the chromosome, right? So now, so cytogenetics was based upon microscopy, a hematologist or a tumor biologist would sit for hours looking at the chromosome. But before you look at the chromosome, you have to do a nice slight preparation so that there is no aberration in the chromosomal structure by doing the experiment, right? So let's see now. See, this kind of questions are very so how many 21 3 mega base pairs, 3100 mega base pairs per haploid genome, 62 mega base pairs in diploid conditions, right? It will double. So you have to, so these statements, these sentences can easily be converted into a question which can trick you. So 3 billion versus 3 mega base pairs, so on and so forth. So this was the way, clinicians, pathologists used to visualize the chromosomes to look for diseases, and diseases were mainly coming from congenital or a cancer that will develop at a later stage. Now, because of the technology, and we talked about sequencing methods in chapter number 2, where chapter number 2, where we talked about PCR, DNA synthesis, sequencing. So sequencing is the essence of all the data that we have. The human genome is so important from evolutionary point of view, and from disease point of view. And there are many diseases that the child or a kid is born with, and then parents think how this happened, right? So, for example, there are some very rare diseases. One of the rare disease is a problem where a child, a newly born child, is born totally normal, but after a few days, child goes into sleep in a sleep stage or coma, and then the samples are sent for genetic analysis, and they figure out that the gene that would synthesize proteins, amino acid genes, are problematic. And because amino acids are not being synthesized, so the kid, the child has a very high level of acidemia in the stomach because of one enzyme, and the pH is changed. So if a person cannot synthesize protein, some of the genes that cannot digest protein, how will this kid consume proteins? So there are special diets for these kids, right? So why this happened? This happened because of DNA sequencing and the knowledge of human genome. And one can go back and see which where did the kid get the mutation from one of the parents and both the parents. In many parts of the world, the marriages happen within extended families. So those can lead to problems. There are many villages, many cities where families marry, and then because the gene becomes very restricted, so if there is a disease, it spreads in genetic disease. It spreads in the whole village, in the whole community. And I have read that there is a place where many male kids in that village, in that neighborhood, have Down syndrome. So because of this data, one can go back and take informed decision, look at the genes, the DNA, and see what the problem is and take care of those problems, and or take corrective measures for future generations, right? So human genome is to know, like you said, I'm going to read your term paper, and I'm going to read hopefully word by word, sentence by sentence, and so if there are mistakes, right? So it needs to be corrected, or and so you should look at those proofreading. So proofreading, if the proofreading cannot happen in human genome when a small kid is born, right? But there are technologies which can do in certain diseases, but in many diseases still. And I remember Moderna that was that it got engaged. So many companies like Moderna, Pfizer, um, who were investing in rare diseases had to shift their focus and to focus on pandemic. So pandemic caused a lot of stress, not only for normal life, but people who had different kinds of pre-existing ailment. So all these companies focused on vaccine generation, which vaccine production. So the reason I'm discussing this with is to look at the technology in a broader way, right? So and human society, yes, if it is a developed country, it is good. But in a developing countries or countries which are not developed, there are a lot of challenges with health. And so, for example, sickle cell anemia, right, where there is one base mutation, and it can crumble the structure of hemoglobin. But people now we know that sickle cell anemia was mainly located in those areas where malaria was more in tropical countries. Malaria was more prevalent. But hopefully we talked about CRISPR. Maybe gene editing is the future for such kind of diseases. And people are traveling, so when they're carrying going to different countries, different neighborhood, and trying to settle down. So gene is the flow of genes or chromosomes or genome is very, very, very vibrant, right? And to realize something after in next generation where you have less time to correct things if the disease is severe. So as I mentioned to you that most of the first generation of human genome sequencing was focused on protein coding genes, right? Now see here, there are various ways, various platforms in which these genes were analyzed, right, by bioinformaticians. And let's look, they got various different numbers. Some of the total numbers, they're close, but some of the numbers are very off. So the knowledge of protein coding genes are important, but there are less variations in the protein coding genes. So let's say if this is the class and there are 23 students or 24 students, including me, 24 or 25, we can look at our hemoglobin gene. And because hemoglobin gene is a protein coding gene, it is possible that all of us genes are very identical. Because protein coding genes are very identical, the changes happens in non-protein coding genes. That is very, very difficult to know. So protein coding, as I mentioned to you about a situation where a kid had a problem with that gene, which had ability to synthesize protein or digest protein. So those are easily trackable with this first generation data of human genome. But the next generation about non-coding DNA was not known. So once you know the protein coding genes using various bioinformatics software, so what is the role of bioinformatics? Bioinformatics is a discipline of biology where biology has collaborated or biology has integrated with computational sciences. And the reason is to analyze large amount of data. Right? And it is very good because in COVID-19 pandemic, we were not allowed to come to the campus and we're not about to go into the labs. I have my lab on the second floor of this AB1 building and I couldn't get the access. So I could learn bioinformatics and I could do research from my home. So the biologist could, and computational people, they collaborated to develop a new branch of biology, bioinformatics, in discipline area. And the reason was to analyze large scale data. Right? So think about, there are 3 billion bases, a little bit more than 3 billion bases in the 46 and the 24 chromosomes, right? If you look at haploid condition in humans. So how to look into it, right? You need a special computer, you need a special server, you need a special analytical tools. So this is a limited picture that you take 20,000 protein coding genes and you can classify these into enzymes, into cellular junction proteins, surfactants, structural proteins like microtubule, storage proteins which are present in Golgi apparatus, transporter, cytoskeletal protein. So all these are different proteins, and they have, if you see here, the maximum proteins. So you can see the percentage. Calcium binding protein is almost how much it is, 0.4%. Immunity protein, 0.6%, right? Transcription factors, large, 12%. Transcription factors helps in transcription. So you can see the distribution, 0.1, 0.1, 0.2, 1.3. So you can imagine that in a given time in a cell, there are thousands of proteins that are working, right? We are working because our proteins are working. If a protein doesn't work, then we will feel the effect. Now, out of these, some genes, some proteins are very important, like BRCA2, BRCA gene, which is, if mutated, it can lead to uncontrolled division and mostly seen in breast cancer. So all these genes have been characterized, and all these data are available for public, for people across the world, right? That's what's the most rewarding part of it, that this Human Genome Project was conceived in 1986. It gave a start in 1990. In 2001, first batch. In 2013, there was an update, and now in 2022. So how many years? 10, 20, 30 plus years. But now you have it on your mouse, in your laptop, in your cell phone. And if you want to look anything, you can go and look. And also, besides these data, there are also data where patient samples are being sequenced and they're putting it on a platform. So information is available. So you can look at your own genome and look at the genes, look at the function of those genes, and try to help a friend or a colleague or someone who has lesser knowledge of biology or health. And remember, I think I began this class this semester by saying that the coming century, including present, it's going to be about health. Health can be affected because of climate, environment, our lifestyle, right? And these climate and environment will have a major impact on our lifestyle, right? So it's very important, to look at the history, because you have to see, when people did not have a machine, people did not have a PCR, people had limited facilities. That was the time when people conceived the idea, right, about DNA. So 1870, and I would go a little bit backwards about Mendel. Mendel was not a scientist. He was not a biologist. He was a naturalist with impeccable knowledge of statistics. He laid the ground of genetics that time by just looking at or monitoring the pea plants and doing the quantitative analysis. So quantitative analysis of chromosomes lineage is genetics, right? Quantitative genetics is what? Quantitative biology, where you try to see how chromosomes is passed from one generation to the second generation. And if everything is normal, then people will not look at it. But if there is a problem, people will go and try to study and analyze his or her own inheritance, where the disease came from. So 1870, Mischer discovered the chemical, had an idea that there's a nucleic acid in the nucleus and it has little bit acidic character, right? So that's how, small discovery, but it was important. And then 1940, genetic material, Avery and McLeod. This was done in New York, Rockefeller University, if I'm not wrong. Then 1953, you saw Watson and Crick. I discussed that paper. Two scientists, Pauling and Fraser, they proposed that the DNA was triple helix, but DNA was double helix. Watson and Crick got insights based upon the work of Rosalind Franklin in UK, Cambridge. And she didn't get the due credit, and also she passed away early. So that's another side of science that you should study, right? You should know about, right? That what's the real story, who are the real contributors, and that would help you to navigate your career, right? To be careful that what work you do, you have the total entitlement. You are the discoverer, you are the inventor, your name should be there and you should benefit. So these are the lessons of, and then in 1965, East tRNA was working. People were, in those times, molecular biology was to grow bacteria and to learn how to manipulate bacteria in 70s and 80s until, sequencing and PCR was discovered in 83, right? So PCR, so when bacteria was there, people were isolating plasmids, people were understanding about restriction enzymes, and then PCR and sequencing happened, and the effort towards human genome was devoted. First draft in 2001, the next generation sequencing, high throughput sequencing. So and technology developed in various directions. If the machines for sequencing developed, improved, then the analytical tools, the bioinformatic tools also keep up pace with the development of technology. Why that was possible? Because scientists collaborate, right? It's not possible that one person can do everything. So in a hospital, a pathology department that gives the report has to do an excellent job, work with the medicine department or pediatrics department, and then the doctors, the surgeon, the nurse, everybody has to collaborate. In a hospital or institution that has this collaborative, effort, they always be ahead of time, and that's why U.S. became one of the leading science, leader of scientific world. Collaborations, and yes, there are problems that we should address. So one of the problem I'm addressing, this Watson and Crick and Rosalind Franklin data, and then in the next class when we talk about patenting, we will talk about HeLa cells, Henrietta Lacks cells, how it was developed, and how scientists use it in all the lab, many of the labs. But the person who donated this tissue, cervical tumor tissue, did not get any credit. So we have to look at that part of the science also. So so each year there was some development, some progress, and it moved from 1990 to 2001 to 2013 to 22 and further because of collaborative effort. Another thing I would like to make a point that Human Genome Project led the collaboration between the government and the private companies. So private company was led by Craig Venter. The name is Celera. I believe this is somewhere in Maryland, and NIH, NIH, and this was funded by, so this was one of the public project that was the outcome of private and public collaboration. So I have already mentioned about the history. These are some of the terminologies that helps to define the chromosomes, right? So genetic markers are present on the chromosomes that give an identity, and genetic markers could be a biomarker. For example, let's say a BRCA gene could be a biomarker for breast cancer. Androgen receptor could be a or PSA, prostatic specific antigen, could be a biomarker for prostate cancer. And so one can easily know based upon this data that in which chromosome BRCA should be looked for. What is the length? How many exons? What is the length? And so on and so forth. So all these information makes genetic markers, biomarkers, genetic map. Yeah? If you go back to Mendel used like homozygous dominant, heterozygous dominant, or recessive, so on and so forth. But now we talk about genetic markers, biomarkers, genetic map, and so what was the technique that was used for sequencing Human Genome Project? Was shotgun sequencing. A genomic DNA, which is 3 billion base pairs, is broken into pieces by physical or by restriction enzymes. And what I want to point you here is that when you break these DNAs randomly, right? So it gives various fragments, and these fragments could be overlapping. Listen to my sentence. These fragments that break, it could be overlapping. And this matching of overlapping helps to sequence the chromosome, because so this chromosome is this much, this is this much, what happens in the middle? So this particular clone would overlap from this end to this end. So you can walk down the chromosomes while putting the sequence. So this is a lot of tedious work, right? It's not easy, but think about it from how people have done this work. Then these fragments, they're cloned in back vectors, bacterial artificial chromosome that I mentioned to you in second chapter, right? And then these are sequenced, and then sequencing methodology has also improved. So a library, so you get these fragments, and from this you make library, and you keep that library and sequence it systematically, analyze it systematically, and see what is the linear arrangement of this DNA, and then corroborate to chromosomes. So broadly, there could be two types of marker. Type 1 marker is coding gene that codes for protein or RNA, right? And they're less polymorphic. As I mentioned to you, that if this is a class and we sequence the hemoglobin gene amongst all of us, it will be almost similar, less polymorphism, unless there is a situation where somebody is having a mutation for sickle cell anemia. Right? So the genes coding for proteins are type 1 marker, and they show less polymorphism. But type 2 marker are from those genes which doesn't code for proteins. And that is feasible now with the sequencing with the latest sequencing data that came in April 2022. This would have been helpful with 2001 data, but 2001 data of sequencing may not be good for type 2 markers. But the results that came out in April, just last month, I believe, will be good for both. So, and also protein coding genes are not repetitive, but there are a lot of repetitive sequences in our DNA. These repeats could be interspersed, not systematic, but some repeats could be very tandem next to each other. And this is seen so interspersed repeats are seen in tDNA, and which compose retropseudogenes, transposons, local repeats. Tandem repeats could be in rDNA and satellite DNA. So I would just touch here about the pseudogenes. What are pseudogenes? So pseudogenes are basically they have all the hallmark of a typical gene that would make protein. They could have promoter, but promoter could be missing. If the promoter is there, the start codon ATG could be missing. There could be frame shift, immature or not premature stop codon, missing intro. So they will look like a gene that is ready to code for a protein, but there will be some kind of defect in this gene, either in promoter, start codon, stop codon, or frame shift, where it will not code for a functional protein, right? So these are called as pseudogenes. So we were here, pseudogenes. And satellite DNA could be microsatellites, mini satellites, and macrosatellites. So microsatellites are short tandem repeats, consist of 1 to 9 base pairs monomer. Tandem repeats could be 10 to 100 base pairs. Microsatellite is rich in adenine and thymine, whereas mini satellites are rich in guanine and cytosine. So microsatellites could be easy to sequence, but the mini satellites could be difficult to sequence because they're very rich in G and C. Similarly, telomeres. Telomeres are also very rich in G and C. And GC rich, they have three hydrogen bonds as compared to AT, which have 2 hydrogen bonds. Okay. And also known as simple sequence repeat or short tandem repeat. Mini satellites, they're called as variable number tandem repeats, VNTR. Right. So these are the types of mini satellites, microsatellites, and macrosatellites. So you can look at the specification. Microsatellites near telomeres, 20 base pairs, 1 to 390 base pair. Macrosatellites are near centromeres. So centromeres is very, very heavily methylated, and where the kinetochore attaches during mitosis or meiosis, there is a small sort of a depth area in the centromere where it attaches. With this, in the last class, last week, this Tuesday, we covered basics of DNA, how it was discovered, what was this, because 1953, molecular biology took a turn with the discovery of DNA. Before that, we were working at the chromosomal level, trying to understand genetic diseases. But after 1953, when we came to know exact positioning sequence of ATGC, we went towards cloning and sequencing, and so we have better knowledge of human genome, and that is mainly to understand our evolution, to understand disease patterns, and so forth. So with this, we finish this chapter. So this was a small chapter. Any questions? So just for students who came at a later stage, and when class had already started, number 1, the new deadline for submission of the term paper, which is those questions, is 19th May, which is the day of your exam. Second, I'm going to go through all the quizzes and midterm until your final exam to make sure there is no discrepancy in grading. And also I will look at the assignments to make sure that, that sometimes if a student did not load or upload the correct assignment, has he or she uploaded with the next assignment? That's what I said. So I will go over these things very carefully. And And yeah, and if you have any questions after you get your grades, then you're free to have a discussion. And but until now, I didn't had any major issue with grading in any of the classes. Any questions, if I could answer?
Professor, this is another question. I didn't hear the attendance, and I just want to make sure I'm
Yeah,
on it.
I am not at my home, where I used to have an Excel sheet. So,
Okay.
but I will. I have taken the picture where there is a list of people who are participating, right?
Oh, okay, okay.
I've taken the picture because
Thank you.
I'm not near my home computer. I'm somewhere outside. So if there are no questions, then maybe we should end this class. You all have a good afternoon and good evening. Stay safe.
Thank you, Professor. Bye. Thanks.
Welcome. Bye.